we are offering the students a choice of the modules to see what is the industry fit. We will advise them as to which set of trainings they should use to become industry ready.
Mandar has actually worked and put up a program for the fellows, and this is going to be a two level. One is actually for working for the clinician part. The other one is for looking at research projects, the clinical trials. So we're going to have two parallel sort of programs, and depending on what you want to build, because they're looking at basic research and clinical research. So we need capacity to take care of both these in parallel.
Dr. Govind Babu mentioned this is divided into two parts, and the goal of the molecular pathology and precision oncology fellowship, what you see on the screen, really is towards creating the PIs and co-PIs who can recruit patients into the trials, design the trials themselves at the clinical end, so that we can maximize enrollment for our patients, because these days access to patients is a critical bottleneck in the West. They're not able to finish their clinical trials. And we have a treatment naive patient pool in India who is ready and does not have access, right? So unless we have the PIs to be able to actually recruit, we are not able to do a good job and move the needle towards earlier phase clinical trials.
This is how, adding to NovaSeq or Illumina instrument, the small Chromium X box, it will give you exactly what is happening in which cell. So this will help clinicians, even clinical trials, why some drugs are failing. So all the lot of information you will get it.
Through translational research, we employ these multi-omics technologies available to us to characterize the various cancers, and then using that information, then design these targeted therapies which can be cost effective.
In order to save time, in order to save the valuable reagents. So there is a new technology, acoustic liquid handling technology, that's called as LabSite Echo. What it does is it does miniaturing of the entire reagent process. So it converts microliter reaction volume to the nanoliter reaction volume. And it doesn't use any pipetting tips. It uses sound technology, sound sensing to transfer the liquid. That's the greatest innovation that I know. It helps scientists to reduce cost on the reagents.
We are into cell biology and proteomics. What product we have it is basically a detection system, optical as well as non-optical. When I say optical, it's a camera-based technology what we have it, and non-optical is the PMT detection system, which is generally called as plate readers. When we say imaging, 3D is on the top, so we can do 3D imaging, you can do organoid imaging, you can do high throughput 3D imaging on a chip-based technology. It's very unique.
And the solutions which we are partnering with VCR is primarily around automated immunohistochemistry and in situ hybridization. In future, we also want to partner in the area of digital pathology, which is a newer field. Also launched a open application programming interface with which we actually want to promote a lot of research and development and third-party integration of those algorithms as well.
But cell therapy would pose very interesting challenges when you go to manufacturing. One is that because it's so personalized, it cannot be scaled up, right? Because there's only one person, one donor, and one patient. So what we have come up with is a concept of scale out, and we have some solutions that how do we do that? How do we scale out instead of doing scaling up?
We have got a very powerful technique for a biomarker discovery, both from an discovery purpose as well as from a targeted approach, which is for when you want to study on the large cohort sets. So that's the part. And in order to facilitate the larger audience,